know?
Everything is okay, Professor.
Mm-hmm. Okay, so today we discuss nervous system. Nervous system is classified according anatomical division into central and peripheral nervous system, CNS and PNS for short. Central nervous system includes brain and spinal cord, and peripheral all the rest, which is nerves, nerve endings, and ganglia. It's plural from ganglion, groups of neurons outside the central nervous system. According to the physiology, we have the functional division of nervous system into somatic and autonomic. Somatic innervates voluntary muscles, skeletal muscles of our body, and autonomic one innervates internal organs, blood vessels, and glands, and is subdivided into sympathetic and parasympathetic. It means that the efferent endings of the neurons of somatic nervous system innervate muscle fibers, and the efferent endings in the autonomic nervous system innervate smooth muscle cells and glandular epithelial cells in glands. In this picture, you can see which organs are innervated by parasympathetic and sympathetic parts of autonomic nervous system. As you can see here, most of the organs are innervated by both sympathetic and parasympathetic nervous system. For example, as we discussed previously, the heart is innervated by both of them, and sympathetic is for the reaction for flight or fight. It increases the rate of heart beating, and parasympathetic vice versa, decreases it. And also, as you can see from this picture, both parasympathetic and sympathetic um, parts of autonomic nervous system include both central portion, which is located inside the spinal cord or in the brain, and peripheral one, which includes nerves and ganglia, the groups of neurons located somewhere inside the organ or near the organ which is innervated by this autonomic nervous system. You should recall everything that you know from the topic about nervous tissue. Nervous tissue makes nervous system, and nervous tissue includes two types of cells: neurons, which are major cells of nervous tissue, and glial cells, which are accessory cells. Glial cells are subdivided into the glial cells of central and peripheral nervous system. In central nervous system, we have macroglia, which includes astroglia, ependyma, and oligodendroglia, and microglia, which is glial macrophages. In peripheral nervous system, we have two kinds of glial cells: Schwann cells and mantle gliocytes. Neurons are classified into four types according to their morphology and the presence and the number of axons and dendrites, into unipolar, bipolar, pseudo-unipolar, and multipolar neurons. As you remember, unipolar neurons have only one axon, and they are present only during embryological development. Bipolar neurons have one axon and one dendrite. They can be found in the sensory organs. They are sensory neurons. Pseudo-unipolar neurons resemble unipolar. This is why they are called pseudo-unipolar, but actually they have both axon and dendrite, but they start as one process and split soon into two, into one axon and one dendrite. Such neurons can be found in the dorsal root ganglia. Multipolar neurons are about 99% of all the neurons, they have usually one axon and many dendrites. They can be found in the brain and spinal cord and also in some ganglia outside the central nervous system. In this picture, you can see different types of glial cells. Here in this frame, you can see two types of the glial cells of the peripheral nervous system, which are Schwann cells, which make the myelinated or unmyelinated nerve fibers in the PNS, and second type, mantle glial cells, which surround cell bodies of the neurons of the peripheral nervous system. All other pictures represent the glial cells in the central nervous system. Here you can see the protoplasmatic and fibrous astrocytes. Protoplasmatic are found in the gray matter and fibrous in white matter of brain and spinal cord. In this picture you can see two of many functions of the astrocytes, such as formation the outer membrane which separates brain from the pia mater, and here the formation of the blood-brain barrier, which protects neurons from something bad from the blood vessels. Oligodendrocytes make myelin in the central nervous system, like Schwann cells in peripheral nervous system. Here you can see three types of the ependymal cells. Choroid ependymal cells, these square cuboid ones, tenicyte, and general ependymal cells. They are associated with the cerebral liquor in the cerebrospinous liquid, and they have microvilli and cilia. Microglial cells are glial macrophages that perform the function of phagocytosis. In this picture you can see the neurons and glial cells in gray matter and the white matter of the brain and spinal cord. In white matter you can see only the processes of neurons, and in gray matter both cell bodies and processes of neurons. The glial cells which are seen here are astrocytes, which have the end feet which surround blood vessels and form the blood-brain barrier here. These are oligodendrocytes, which make myelinated sheets around the axons of the neurons, and this one, small one, is the microglial cell. Here is the ventricle of the brain surrounded with the ependymal cells. In this picture you can see the diagram of the blood-brain barrier. This barrier is shown with this bracket, red in color, and it includes three major elements. The cytoplasm of the endothelial cell here, next basement membrane of the endothelial site, and third component, the astrocytes and feet, which surround this blood capillary. Here we have the blood capillary of type 1, and this blood-brain barrier is semi-permeable. It means that it performs the transportation of only some chemicals, not all the chemicals. It regulates transfer of chemicals from blood to neurons, and so it protects neurons from all the bad stuff that can come here from blood, such as toxins, bacteria, and all unwanted here chemicals. Here in this picture we can see the choroid plexus, the structure which includes in its structure the blood capillaries surrounded with loose fibrous connective tissue and covered with one layer of cuboidal cells called choroid ependymal cells. Here you can see the part of this choroid plexus at high magnification, So we see here blood capillary with some erythrocytes inside. It is surrounded with loose fibrous connective tissue and one layer of cuboidal cells lying upon the basement membrane. Between these cells we have the number of cell junctions, including tight junctions. And the functions of these choroid ependymal cells is to form the blood-liquor barrier, the barrier between the blood and cerebrospinal fluid or liquor. So the liquor, the cerebrospinal fluid, which is located inside the brain ventricles and the central canal of the spinal cord, is made by the choroid ependymal cells from the blood plasma as a result of filtration of blood plasma through this blood-liquor barrier. So the cerebrospinal fluid or liquor is made by the choroid ependymal cells from blood plasma. These choroidal ependymal cells are located in the choroid plexus, which is located inside the ventricles of the brain. This liquor is then circulating inside the ventricles and inside the central canal of spinal cord, and also in the subarachnoid space around the brain and spinal cord. And finally, the liquor goes to the venous circulation through the arachnoid granulations. So here you see this subarachnoid space between the pia mater and arachnoid, and the cerebrospinal fluid from here goes to the sinuses and then into veins. Development of nervous system. Nervous system develops from the ectoderm, which in the central its portion develops into neuroectoderm, unlike the other part which will give the skin ectoderm. So neuroectoderm then gets thicker and forms the neural plate. Then it makes invagination and forms the neural groove, and then neural groove forms the neural tube and neural crest cells. This process starts from day 18 of the embryonic development, when we have the neural plate formation, then neural groove, and it forms neural tube and neural crest cells. The neural tube will give rise to all the neurons and glia of the brain and spinal cord of the central nervous system. The neural crest cells will give rise to the neurons and glia of peripheral nervous system, which means the autonomic and sensory ganglia and sensory cells and cells of pia mater and arachnoid mater of the brain. Also, we have the third embryonic source of development for the nervous tissue. They are ectodermal placodes. Placodes means thickening of the ectoderm in the frontal part of the embryo, and these thickenings of ectoderm will give rise to the ganglia of cranial nerves 3, 7, 9, and 10. In this picture you can see the neural tube. Neural tube has layers. The innermost layer is called ependymal layer. Here we have the proliferation of neural stem cells, which will further give rise to neurons and glia of central nervous system. Then those proliferating cells migrate to form the next layer, mental layer. Here we have neuroblasts and glioblasts, the cells during their development into neurons and glial cells respectively. When neurons mature, they form first only one axon, and they are so-called unipolar neurons, and the axons grow towards the periphery of the nervous tube and form the outermost layer of the neural tube, which is called marginal layer. So marginal layer is composed of axons of unipolar neurons. Those axons go outside and try to find their target, for example, muscle fibers. If they cannot find their target, they die with apoptosis. During development, the cells which form first the neural tube in the area where the spinal cord develops will form the gray matter, which will surround the central canal of the spinal cord. And the area first formed by the axons of unipolar neurons, which is called marginal layer, will next form the white matter of the spinal cord, which is composed of the axons of neurons. In the frontal part of the neural tube forms the future brain, and during the formation of the brain, the new neurons are born and they start to form the connections between them, and this process, the formation of contacts, which is called synaptogenesis, the formation of the synapses, this process continues throughout all the life, and the formation of new neurons is possible only during embryonic development. After birth, no new neurons can be formed, but new synapses can be formed till the end of life of the person. Now we will discuss the reflex arc. The reflex arc includes the two or more neurons. It starts with afferent or sensitive nerve endings of the receptor sensory neuron, which is called afferent neuron. Its dendrites end with receptors, which are subdivided into free and non-free endings. Non-free are subdivided into encapsulated or not encapsulated, depending on the presence or absence of the surrounding the dendrite, the glial and connective tissue cells. So if we have just the free dendrite, it will be called free nerve ending. If the dendrite end is surrounded with glial cell, it will be called non-encapsulated. If we have the surrounding glial and connective tissue cells, it will be called encapsulated. So it is the classification of ends of dendrites of sensory neurons. So impulse is generated here in the receptor, then impulse goes to the cell body, the perikaryon of the sensory neuron, then through its axon to the second neuron, which is called interneuron or relay neuron. Between neurons we have the connection called synapse, and the impulse in synapse goes from axon of one neuron to dendrite or perikaryon of the second neuron. Then through the axon of the second neuron, the impulse goes to the third neuron, the motor or effector neuron, and finally through the axon of this motor or effector neuron, impulse goes to the target organ and ends with motor nerve ending or motor plate in the case of somatic reflex arc, where we have the connection of the axon of the motor neuron with the muscle fiber of skeletal muscle tissue. Or if we talk about the autonomic nervous system, when we have the connection between the axon and the smooth muscle cell or the glandular epithelial cell, we will have the secretory nerve ending. Here in this picture, very important, that you should know, we have three types of reflex arcs: A. Somatic reflex arc, B. Sympathetic, and C. Parasympathetic reflex arcs. You should know the structure of these reflex arcs, and you should be able to explain the difference between them. In this picture, we have the simplest example of the somatic reflex arc, which includes only two neurons. One sensory neuron, and second one is the motor neuron. We can have also intermediate interneuron relay neuron, which cell body will be located in the posterior horn of spinal cord. So this reflex arc starts with the receptor, the end of dendrite of the pseudounipolar neuron located inside the dorsal root ganglion. Then the information goes to the interneuron or directly to the motor neuron, which cell body is located in the ventral horn of the spinal cord. And the axon of this motor neuron goes to the periphery, to the innervating muscle fiber, and ends with the efferent ending called here motor end plate. Next, sympathetic reflex arc also starts with the receptor, which is the end of dendrite of the pseudounipolar neuron located in the dorsal root ganglion, the same as in the somatic reflex arc, but its axon goes to spinal cord and ends on the interneuron, which is located in the lateral horn. Then the axon of the interneuron goes outside from the spinal cord to the sympathetic ganglion, marked here with this circle. The ganglion is the group of neurons inside this peripheral nervous system. One neuron, second neuron are shown here, and the axon of the interneuron ends with synapse with the cell body or dendrites of the third efferent neuron located in this sympathetic ganglion. Axon of the efferent neuron goes outside to the innervating organ. Here, smooth muscle cells are shown as target tissue, and so here we have the secretory efferent endings. These are the varicose thickenings of the axons containing neurotransmitters, and when impulse goes to the secretory nerve ending, the neurotransmitters are released from these varicosities and act upon the smooth muscle cells, forcing them to contract. So in the case of the sympathetic reflex arc, we have the sympathetic ganglion, and the third efferent neuron is located not inside the spinal cord but in the sympathetic ganglion. Number 3 is for the parasympathetic arc. It starts the same as the receptor, the end of the dendrite of the pseudounipolar neuron located in the dorsal root ganglion. Its axon goes to spinal cord, to the intermediate zone, and as in the sympathetic one, the axon of the intermediate axon goes outside from the spinal cord to the ganglion. But the ganglion, unlike in the sympathetic reflex arc, is located very far from the spinal cord inside the target organ, inside the layer of smooth muscle cells. Such ganglion located inside the target organ is called intramural ganglion. In some cases, such ganglia can be found not inside the organ itself, but very close to the innervated organ. So in this case, we have the intramural ganglion, and here in this ganglion, the third neuron, the efferent neuron, is located, and its axon goes outside and innervates the tissue, the smooth muscle tissue, and Also, we will have here the secretory nerve ending, like in the sympathetic reflex arc. So, the difference between the sympathetic and parasympathetic are in the location of the ganglion. This is the major difference. We have other differences, but this is the major one. In sympathetic ganglion, we have the in sympathetic reflex arc, the ganglion is located closer to the spinal cord. And in the parasympathetic one, the ganglion is located far from the spinal cord, close to the target organ or inside the organ. And then it is called the intramural ganglion. So we will discuss now these reflex arcs in details. So somatic reflex arc, first neurons, pseudounipolar neuron located in the dorsal root ganglion. It has dendrite which goes to the periphery and ends with receptor. So impulse goes this way along the dendrite, then along the axon. It enters the spinal cord and ends with synapse with the interneuron. Axon of interneuron innervates the motor neuron, which is located in the ventral or anterior horn of the spinal cord, and the axon of motor neuron goes outside from the spinal cord through anterior root and goes to the skeletal muscle fiber and ends here with the motor end plate. Also, what is important, you should know the neurotransmitter released by the efferent neuron in every reflex arc. So in the somatic reflex arc, we have the acetylcholine released by the motor neurons and innervating skeletal muscle fibers. Sympathetic reflex arc also has the first sensory neuron inside the dorsal root ganglion, the pseudounipolar neuron, which ends with receptor. Impulse goes through dendrite, then through axon of this neuron. Axon of pseudounipolar neuron enters the spinal cord and ends on the interneuron located in the lateral horn, unlike the previous reflex arc where interneuron is located in the posterior horn. So here interneuron is in the lateral horn. Its axon goes outside the spinal cord and goes to the sympathetic ganglion. In this example, the ganglion of the sympathetic chain. Here we have the third neuron, effector neuron, and its axon goes to the innervated organ. The muscles, smooth muscles of intestine, shown here as an example, and ends here with the nerve ending of secretory type. The neuron transmitter released here is the norepinephrine. We have two types of sympathetic ganglia, paravertebral, which are sympathetic chains are located to the left and to the right from the spinal cord, and second type, prevertebral ganglia, which are celiac, superior mesenteric, and inferior mesenteric ganglia. Now, parasympathetic reflex arc has two divisions, sacral and cranial divisions. First, sacral division of the parasympathetic reflex arc also has the first sensory neuron in the dorsal root ganglion. Its axon goes to the spinal cord, to the parasympathetic sacral nuclei in the intermediate area of the gray matter of the spinal cord. The axon of this interneuron goes outside to the innervating organ and ends on the third effector neuron located in the intramural ganglion inside the target organ. The axon of this efferent or effector neuron ends with the nerve ending of secretory type, innervating smooth muscles or glandular epithelial cells here. The neurotransmitter here is acetylcholine. And the cranial division of the parasympathetic reflex arc has the sensory pseudo-unipolar neuron not in the dorsal root ganglion, but in the cranial ganglion of the cranial ganglion, located not in the spinal cord but in brain. This is why it is called the cranial division. Its axon goes to the second neuron located also in the brain. This particular arc is for the nucleus vagus, for the vagus nerve, and here we have its interneuron, which is located in the nucleus dorsalis for nucleus vagi, in the rhomboid fossa. And the axon of this interneuron goes to the periphery and forms actually the vagus nerve and goes to the target organ, where it ends on the third neuron inside the intramural ganglion. And also we have here the acetylcholine as the neurotransmitter of the effector neuron. Why we have two types of parasympathetic nerve arcs is clearly evident from this picture. As you can see here, some portion of the parasympathetic nervous system has its central part in the brain, so these will have the will consist the cranial division of the parasympathetic nervous system, and here we have the sacral division, where the central portion is located in the sacral part of the spinal cord. So the parasympathetic reflex arcs, the cranial division, innervate the organs of the head and neck, and as you see here for the vagus nerve, it innervates almost everything in our chest and in the abdominal cavity, such as heart, lungs, stomach, liver, and intestines, including the proximal part of the colon. And the sacral division of the parasympathetic nerve system innervates the distal part of the colon, urinary bladder, and gonads. Intramural parasympathetic ganglia are the groups of neurons located inside the innervated organ. This is the intramural ganglion found inside the heart wall, in the epicardium of the heart. You see here a lot of adipose tissue, so this is the epicardium of the heart. This is one neuron, second neuron, third one. They have large cytoplasm, basophilically stained, large light nucleus, and well-developed nucleolus inside it. All other cell nuclei here are the glial cell nuclei and the cell nuclei of loose fibrous connective tissue. Here we have another example of intramural parasympathetic ganglion. This is the ganglion inside the intestinal wall. It is located between two layers of muscle layer of the intestine. One muscle layer is here, second layer is here, and between them we have sandwiched ganglion with large neurons black in color in this example because of staining, which was silver impregnation and hematoxylin. Now, nervous center. This is an important term that you should know by heart. So, nervous center is a group of multipolar neurons that are located in some certain site within the central nervous system. Such neurons have the same origin and they perform a common function. Nervous centers are the sites of neuronal integration. It means that interaction and information transfer between the neurons, which implies the presence of synaptic transmission. So, a group of neurons can be called a nervous center if it this group of neurons contains multipolar neurons, and if we have synapses between those neurons. For example, dorsal root ganglion cannot be called a nervous center because it consists of pseudo-unipolar neurons and not multipolar, and we have no synapses and no transfer of information between the neurons. So nervous centers are subdivided into three types: the ganglia, the groups of neurons outside the central nervous system. They are sympathetic and parasympathetic ganglia, which we have seen before in heart and in the intestine. Second type, nuclear type, the same but not in the peripheral but in central nervous system. So it is the group of neurons inside central nervous system, inside brain or spinal cord. Spinal cord is shown in this picture. And the third one type is the plane or screen type of the nervous center. It is located in cerebellar cortex and in the cerebral cortex in the brain. So here in this picture we have the cerebellar cortex, where the neurons are located in layers. They are not in groups but in layers. And here this layer of neurons, of the largest neurons, form the plane or screen type of the nervous center. The brain and spinal cord are covered with the meninges and are located inside the neurocranium and vertebral column that protect them from injury. So we will discuss the meninges, which include dura mater, arachnoid mater, and pia mater. They are Latin terms. The closest to the brain and spinal cord is the pia mater. So here we have the spinal cord in cross section. The pia mater is the closest. It covers the brain and spinal cord tissue and goes to all the sulci. It consists of loose fibrous connective tissue and squamous cells. Here we also see it. Next goes arachnoid matter. Here it is. It doesn't go to the sulci. And the outermost is the dura mater, which consists of dense fibrous connective tissue and also squamous cells. It is found here. The same is for the brain. Here we have the brain cortex. It is covered with the pia mater, blue in color, which goes into all the sulci. Next goes the arachnoid mater. Here is it, and the dura mater. Dura mater is the closest to the skull bone, to the periosteum of the skull bone. Now we go to the structural organization of the spinal cord. The spinal cord has the gray matter, which contains cell bodies of neurons in the shape of the butterfly wings, and the white matter, which surrounds the gray matter. White matter is composed of nerve fibers, which go along the spinal cord. So in the cross section of the spinal cord we will have the cross sections of nerve fibers in white matter. The axons of the sensory neurons enter the spinal cord through the dorsal root, and the axons of the neurons of the gray matter of the spinal cord exit spinal cord with the ventral root. The same is in this diagram. So here we have the gray matter, which includes cell bodies of neurons, And here is the white matter, which includes the cross-sected nerve tracts, nerve fibers. Here we have also dorsal root ganglion filled with pseudo-unipolar neurons, which dendrites go to the periphery and axons go inside the dorsal root of the spinal cord to the gray matter of the spinal cord. And here we have the anterior root of the spinal cord, which has in its structure the axons of neurons, which cell bodies are located in the gray matter of the spinal cord. These can be the axons of the neurons of the lateral horns for the autonomic reflex arcs, or axons of motor neurons, which cell bodies are located in anterior horns for somatic reflex arcs. So in all cases, the axons go from the spinal cord inside the anterior root. The dorsal and anterior root meet together to form the spinal nerve, which is mixed nerve because it is formed both by the dendrites and axons. This is the picture that you should use as the example to fill your picture in the album about the spinal cord. So all the labels that are present here, you should know them and you should use them for your picture. And this is the real histological preparation, in which we see the gray matter in form of butterfly wings, white matter, the central canal, and dorsal root ganglion also is seen here. Dorsal root and ventral root are also present in this picture. And also we have here the meninges. The pia mater will be found here, arachnoid and dura mater. Here is our histological preparation of the cross-section of spinal cord with silver impregnation. You should know the difference between anterior and posterior horns in histological preparation. They are such as the anterior horns are shorter and wider than posterior, which are longer and thinner. And anterior horns contain the largest neurons, seen also in this small magnification, low magnification. So here we see this preparation at high magnification. This is the central canal. This is the ventral root, dorsal root. In the gray matter, we have cell bodies of multipolar neurons. They are seen here, cell bodies of multipolar neurons. Also, we will have here axons and dendrites of these neurons. Also, glial cells such as astroglia, oligodendroglia, and microglia, and blood vessels. Here we have the central canal surrounded with ependymal glial cells. In white matter, we have the cross-section of myelinated nerve fibers, which look like small white circles. Also, glial cells such as astroglia, oligodendroglia, and microglia, and blood vessels. Here you see these groups of neurons, which are called nuclei. These groups of large multipolar neurons in the ventral horn of spinal cord form motor nuclei. They contain multipolar neurons, which are motor neurons. Their axons will go outside from the spinal cord and innervate the skeletal muscle fibers. At the highest magnification, we see here in silver impregnation preparation the neurons with their pericardia and the processes. Both axons and dendrites are seen with this preparation. Glial cells are not very well seen, and all the space between the neurons and glial cells is filled with the so-called neuropil, which is the network of neuronal and glial processes. So we have no extracellular matrix in the brain and spinal cord because all the space is filled with the processes of neurons and glia. Only small amount of tissue fluid can be found between those processes. So you should remember the types of staining for the nervous tissue. The major two are silver impregnation and Nissl staining or methylene blue staining. The silver impregnation reveals neurofibrils, which is the result of fusion between microtubules and intermediate filaments, the cytoskeleton elements, during this histological preparation. And these neurofibrils are found both in the perikaryon and in all the processes. So we see the cell body and all the processes of neurons. Methylene blue stains Nissl bodies, which are the clusters of rough endoplasmic reticulum and polyribosomes in the cell bodies and the proximal portions of dendrites. So axons are not seen in this histological preparation. These are the nuclei of glial cells. Here we see the cross-section of spinal cord with methylene blue staining. Anterior median fissure in this particular picture is above the central canal, so the anterior horn will be in the upper portion of the picture and the posterior horn is in the lower portion. The central canal is surrounded with the ependymal glial cells. The cell bodies of neurons form the gray matter, and this is the white matter, inside which you see these cross-sected myelinated nerve fibers. At high magnification, we see here central canal filled with cerebrospinal fluid, and central canal is surrounded with ependymal cells, which have cilia upon their apical surface, and these cilia, they are motile, they can move, and these cilia help to move the cerebrospinal fluid in the central canal. These are cell nuclei of glial cells, and these are the neuropil processes of neurons and glial cells. It's a gray matter at high magnification because here we see the cell bodies of neurons, and these processes are dendrites because axons are not seen in this type of staining. These small nuclei are the nuclei of glial cells. They can be nuclei of astroglia or oligodendroglia or microglia. We cannot define between them. This is the blood capillary, and this is the neuropil. At high magnification, we see the cell body of a neuron filled with Nissl bodies, and dendrites are seen here. Neuropil, nuclei of glial cells. Now, important terms, conducting tracts. Conducting tracts are the anatomically and functionally connected neurons, and through these chains of neurons, the impulse goes in one strictly defined direction. We have three types of conducting tracts: afferent or ascending, which means going upwards, associative tracts, and efferent or descending, which means going downwards, conducting tracts. So afferent or ascending tracts, they transfer impulse upwards from the receptor and through the chain of neurons, one neuron, second neuron, etc., to the highest nervous center. The highest nervous system, center in our body is the cerebral cortex. There, in cerebral cortex, these impulses are processed, and the cerebral cortex decides what what to do next with this synapse, and gives its answer. Associated tracts provide links between the nervous centers, for example, between the cerebellum and cerebral cortex, between the areas of cerebral cortex, etc. So, associative tracts are located in brain, inside the brain. Efferent or descending tracts, they ensure conduction of information downwards from the highest nervous center, from cerebral cortex downwards to the target organ, to some muscle. All the tracts consist not of one, but usually of many neurons. Only first neuron in the ascending or the afferent tract is sensory. Actually, sensory is only this neuron. All other neurons will be interneurons in this afferent tract. So this is very important because we have here some terms usually not the same in anatomy, physiology, and the histology. So the tract is called afferent, but only first neuron is really afferent. Other neurons in this tract will be interneurons. In associative tracts, all the neurons are associative or interneurons, so here it is simple. In efferent or descending tracts, only the last one neuron is really efferent. All other neurons will be interneurons. And now we go to the organs. First organ to discuss will be cerebellum. Cerebellum is located here. It is shown here. It includes in its structure the cortex, which covers it from the outside, and nuclei, which will be located in the central portion, surrounded with white matter. The cortex, which covers the cerebellum and the cerebrum, also have these folds, which are called gyri, one gyrus, many gyri, and invaginations, which are called sulci, one sulcus, many sulci. Here the development of the cerebellum is shown, so it develops from the neural tube, which forms the fourth ventricle, and in its ependymal layer the cells proliferate, then they migrate, and they are so multiples, so that they start to form such foldings. The function of the cerebellum, which means little brain, is to coordinate movements. It controls posture, balance, and fine motor movements, and it is involved in motor learning. The cerebellar cortex, which is shown here, performs the operations of comparing nerve impulses coming from various parts of central nervous system, and it inhibits the activity of the cerebellar nuclei. The cerebellar cortex consists of 3 layers, and all the neurons here will be multipolar interneurons, because we are in brain, and all the neurons in brain and spinal cord are multipolar interneurons. So here is the microanatomy of the cerebellar cortex. So this thin white space is the space between two gyri. This is the sulcus, and the pia mater goes inside the sulcus. It brings here blood vessels, which will go further into the tissue of the gray matter. A yellow bracket shows the gray matter, which includes in its structure cell bodies of neurons, and here white matter, which consists of the processes of neurons which form nerve fibers. So gray matter of the cerebellar cortex includes three layers. The first, the outermost layer, is the thickest layer. It is called molecular layer. It has few cells in its structure. Next goes the Purkinje cell layer or ganglionic layer. It includes one row of very large Purkinje cells. They have very thick and long processes which are dendrites, which go inside the molecular layer. And third layer is called granular layer. It consists of very a high amount of very small cells. The same is shown here in other type of staining. Molecular layer has few cells and almost all the space is filled with their processes. Ganglionic cell layer consists of one row of large Purkinje cells with their dendrites going upwards, and the granular cells consist of large amount of very small cells. And below the granular layer, the white matter is located, which includes nerve fibers. Here we have the plane or screen type of the nervous center, uh, made up with a ganglionic cell layer, because these cells form the answer of the cerebellar cortex. When the impulse goes towards the cerebellar cortex, is processed here, and the Purkinje cells are those cells which give the answer. So this layer forms the plane or screen type of the nervous center. Here we have the general principle of working of any nervous center. So in any nervous center, we have the input of information, the processing of this information, and output of information. Information in our nervous system is present in the form of nerve impulses, which travel along the dendrites and axons of neurons. So input of information forms the so-called afferent pathways. Afferent here means incoming information, and here we will have two types of nerve fibers which bring information here to the cerebellar cortex. The cells of the cerebellar cortex itself perform the processing of this incoming information, such as convergence and divergence of nerve impulses, inhibition and excitement, feedback mechanism, and summation of information. You should learn this from physiology course. And finally, the Purkinje cells, these large neurons, give the answer, the output of information. So the axons of Purkinje cells will form the so-called efferent pathways, which here means the output of information. And this is very important. You should learn this by heart. The diagram of the cells and their network in the cerebellar cortex. So we will discuss it, and it will be very easy for you as a result. So we start with the incoming of information. We have here two types of the afferent nerve fibers, which means which bring here the incoming information. Two types of such fibers are climbing fibers, green in color. They are they bring impulses from the nucleus of the lower olive along the olive cerebellar pathways. So they are the axons of the neurons, which are located in the olive. So these climbing fibers enter the cerebellar cortex, go through the granule layer upwards to the layer of the Purkinje cells and end upon the dendrites of Purkinje cells, and they excite dendrites of Purkinje cells. The second type of the afferent fibers are MOSI fibers. They bring impulses from the vestibular nuclei and from the nuclei of the pons and spinal cord. So these are the locations of the cell bodies of neurons which axons make the MOSI fibers. So MOSI fibers go into the cerebellar cortex and in the granular layer, and they excite the dendrites of the granule cells, which are small cells filling the granular layer. Next, we have discussed two types of the afferents here. Next, we go to the efferent. Efferents, which are outcoming fibers, they bring information from the cerebellar cortex. They are the axons of the Purkinje cells, and these axons carry away the impulses and they end upon the cell bodies of neurons located in the cerebellar nuclei. Axons of the Purkinje cells inhibit the cells of the cerebellar nuclei. So we discussed afferents and efferents, and now we should see what happens inside the cortex itself, how the information is processed here. So the most simple circuit will be this one with the climbing fibers, which innervate the Purkinje cells directly. Purkinje cells become excited, and the impulse goes through their axons to the target neurons in the cerebellar nuclei. And the second pathway is not so simple. Here, MOSI fibers excite the granule cells, then the granule cells, which have very long axons which go upwards through the ganglionic layer, through the molecular layer, to the surface of the cerebellar cortex. Then, here, the axon splits into two portions. One goes one way, second goes other way, and as you see here, the axon of the granule cell innervates all the other cells found in the cerebellar cortex. And as you see these signs, all the axons of granule cells excite all the other cells. This plus means exciting impulse. So granule cell excites all the cells here. Now only Purkinje cell will make the answer of the cerebellar cortex and will make the efferent. All the other cells here, which are Golgi cells, basket cells, stellate cells, they are inhibitory cells inside the cortex itself. They just help to process the information. You should be able to name all the layers of the cerebellar cortex and name the cells located in each layer. And also we have this diagram without any labels. You should be able to use this diagram to explain the function of every cell. It will look like this. So in the granular layer we have granule cells and Golgi cells. In Purkinje cell layer or ganglionic layer we have Purkinje cells. In molecular layer we have basket cells and stellate cells. Now, about the functions of the cells. You should look to these signs. For example, if I ask you about the function of the basket cell, you explain that basket cell receives the incoming exciting information from the axon of the granule cell, and its axon makes the axosomatic synapse with the cell body of the Purkinje cell and inhibits Purkinje cell because of this sign of minus. Another example, what is the function of the stellate cell? The stellate cell receives incoming exciting impulse from the axon of the granule cell, and the stellate cell innervates its axon innervates the dendrite of the Purkinje cell and makes inhibiting impulses through the axodendritic synapse between the axon of the stellate cell and dendrite of the Purkinje cell. So only granule cells in the cerebellar cortex are exciting. So only their axons make exciting synapses. All other neurons here are inhibitory. Golgi cells are inhibitory, basket cells are inhibitory, stellate cells are inhibitory, and also Purkinje cells are also inhibitory. In this picture you see the three-dimensional arrangement of the cells of the cerebellar cortex. You see that the dendritic tree of the Purkinje cells looks not the same in different planes of sectioning. But in general, you see here the same picture. So here we have two types of afferent fibers, afferent mossy fibers, which end on the dendrites of the granule cells, and the afferent climbing fibers, which end upon the dendrites of the Purkinje cells. The axons of the granule cells go to the surface of the cerebellar cortex of the molecular layer, split into two, and these branches of the axons go in opposite directions and form the so-called parallel fibers, which go parallel to the sulcus of the cerebellar cortex and innervate all the cells here, making the exciting impulses. Here you see how the basket cells make the baskets, which are axons which surround cell bodies of the Purkinje cells to make more synapses with the cell bodies of Purkinje cells. This is the histological preparation, cerebellar cortex with silver impregnation. This is why it looks brown in color. What we see here, this white space is the space between the gyri. This is the sulcus, inside which we see the pia mater. Pia mater, which covers the brain, and it has blood vessels in its structure, and these blood vessels will feed the brain tissue. Here is the cortex itself. It is the gray matter containing cell bodies of neurons, and in the center here we have the white matter. It contains nerve fibers. The cortex, the gray matter, contains three layers. Outermost is the molecular layer, then goes ganglionic cell layer with one row of large Purkinje cells, and then goes granular layer. Granular layer is the closest to the white matter. At high magnification, what we see here, this is the surface of the cortex, and this is the sulcus and the pia mater inside it. So this is the molecular layer. Here we have few cells. Most of the space is filled with the processes of the cells. Next goes the ganglionic layer with the Purkinje cells and their dendrites, which go to the surface of the cortex. And a granular layer contains a very high amount of very small granular cells. And next goes the white matter. So these cells, which are located at the uppermost part of the molecular layer, should be the stellate cells. Also in the molecular layer, but closer to the Purkinje cells, here these should be the basket cells. Basket cells make baskets with their axons. Their axons wrap around the cell bodies of neurons, of Purkinje cells, making lots of the synapses with the cell bodies of Purkinje cells. And so these wraps of axons of basket cells are called baskets. This is why such cells are called basket cells. These are Purkinje cells. Here we have their dendrites. Axons should go this way through the granular layer to the white matter. They are usually not seen. And now here in granular layer, most of the cells are granular cells, but some of them can be Golgi cells. Another example of this histological preparation, molecular layer with stellate cells here and basket cells here. Next, ganglionic cell layer with Purkinje cells, cell bodies and dendrites. Next, granular cell layer with the granular cells and Golgi cells. And here, white matter with the nerve fibers and glial cells. At high magnification, here we see large Purkinje cells, they thick dendrites, and the baskets around cell bodies of Purkinje cells made up with the axons of the basket cells located somewhere here. Granular cells are seen here. Another example of the preparation of the cerebellar cortex is the section stained with hematoxylin and eosin. Again, we have here the gyri and sulci, the pia mater with blood vessels is seen here, cerebellar cortex here, and white matter here. In cerebellar cortex, the outermost layer is the molecular cell layer. It looks lighter because it contains few neurons. Then, not very well seen layer of Purkinje cells, ganglionic layer, and granular layer. It is more darkly stained because here we have lots of cells, and white matter. The same at high magnification, molecular layer with few cells. These should be stellate cells. In this area here, basket cells. Here, ganglionic layer with large cell bodies of Purkinje cells. Their dendrites are not very well seen, but their cell bodies are seen quite good. And granular cell layer. Here, white matter. Now, cerebral cortex. Cerebral cortex. Here, in the frontal section, it is seen here, this darker outer line, and white matter below the cerebral cortex. As the cerebellum, the cerebral cortex forms the gyri and sulci, and the cerebral cortex develops from the so-called cortical plate. So neuroblasts, baby neurons, are generated in the ventricle zone, of the developing telencephalon, the frontal part of the brain. And these neuroblasts start to migrate upwards from the place where they were generated along the long processes of the so-called radial glial cells. Here are the radial glial cells with their long processes. Here they are in green color, long processes. And the neuroblasts go along these processes upwards to the periphery to find their final location. They will be finally the neurons forming 6 layers of the cortex. First, the neuroblasts which form future first and 6 layers come to their final position. Next, neuroblasts which go after them, they will they will be placed between those first to form the future second, third, fourth, and fifth layers. Here is the important information: the structural organization of the cerebral cortex. It is organized into 6 layers with no very good seen borders between those layers. The outermost layer, closer to the surface of the cerebral cortex, is called the first or the molecular layer. It has very few cells, the same as in the cerebellar cortex, also molecular layer which contains very few cells. Next goes the external granular layer, or layer number 2. Then goes the pyramidal layer, or layer number 3. Next, internal granular layer, or layer number 4. Next, ganglionic layer, or layer number 5. And finally, multiform cell layer, or the layer number 6. The general feature of this cerebral cortex is the presence of the specific neurons, specific in their shape, which are called pyramidal neurons. They are present in most of the layers, and they have different size from the smallest to the largest. Largest are found in the ganglionic layer. Here they are largest cells, and they are triangular in shape. Triangular in shape in the histological preparation, but pyramidal in three-dimensional view of the cerebral cortex. Here is the cytoarchitecture of the cerebral cortex when we see the cell bodies of neurons, and here is the organization of nerve fibers. You see that, for example, molecular layer, which has very few cell bodies, is filled mostly with nerve fibers. And most nerve fibers are organized into the plexuses. Some of them go along the surface and make tangential fibers, and some go from the surface to the white matter and form the radial bundles of fibers. We have different areas in our cerebral cortex depending on the function of these areas. For example, we have the motor cortex, which serves for motor function to coordinate motor movements, and we have the sensory cortex, which is for the processing of information which comes from the sensory organs such as eyes and ears, etc. And in histology, we have the difference in the structure between the motor cortex and sensory cortex. The motor cortex is called the cortex of agranular type, and sensory cortex is called the cortex of granular type. The difference is in the difference in thickness of the layers. In the sensitive cortex, the second layer, layer 2, and layer 4 are much thicker than in the motor cortex. Compare the thickness of layer 2 here and here. The same for layer 4 here and here, because these layers are processing the incoming information from sensory organs. The motor cortex has very thick layer 3 and layer 5. In layer 5, we have the neurons which may end upon the motor neurons in the spinal cord. But the motor cortex has thinner layer 4 and layer 2. So the thickness of layers depends on the function of the type of the cortex. Important information, this diagram is present in the diagrams for colloquiums, and here you should know the names of layers: molecular layer, outermost; second layer, external granular layer; third, pyramidal layer; fourth, internal granular layer; fifth layer, ganglionic layer; and sixth, multiform cell layer. And also you should learn all these types of neurons. Actually, we have about 100 of different types of neurons in the cerebral cortex, but you should know at least these from 100 of types. This is very important. So, in molecular layer, we have horizontal cells of Cajal, these. In second external granular layer, we have small pyramidal cells and stellate cells. In pyramidal layer, we have the medium pyramidal cells. In layer 4, we have we have the spiny stellate cells. In ganglionic layer, we have the giant pyramidal neurons or Betz cells. They are very, very large pyramidal cells, triangular in shape. And in the multiform cell layer, we have the cells with different shape. This is why this layer is called multiform. Many different forms of cell bodies, and some of them are spindle-like multiform neurons. Almost in all the layers of the cortex, we have the triangular in shape pyramidal cells. You should know at least the pyramidal cells of the layers 2, 3, and 5. They differ in size. In the layer 2, we have small pyramidal cells. In layer 3, medium pyramidal cells. In layer 5, giant or very large pyramidal cells. All pyramidal cells are exciting, and all pyramidal cells make the output of the information. So their axons form the efferent outcoming fibers. Also, these stellate cells are important. They are also exciting. So all pyramidal cells and spiny stellate neurons of layer 4 are exciting. All other types of neurons are inhibiting neurons. As in any other nervous center, in the cerebral cortex, we have the information input through the afferent fibers, information output through the efferent fibers, and processing of information with the help of all the cells which are located here. So, this very important. This is also the task in your album. The cortical connections, the afferent and efferent fibers. We have 2 types of afferent incoming fibers and 4 types of efferent outcoming fibers. The afferent incoming fibers are these cortical fibers. They are called so because they went from cortex and came here also to cortex. So these nerve fibers, cortical fibers, are formed by the axons of the pyramidal neurons of other parts of the cortex. They went here from the white matter, they enter the cortex, and they go through all the layers of the cortex to the surface, to the molecular layer, and here in molecular layer they split in 2 branches, and these branches go along the molecular layer. And while they go this way, they innervate and excite all the pyramidal neurons in the cortex. So cortical fibers came here to this particular part of the cortex from some other part of the cortex. Next, second type of afferent incoming fibers, thalamocortical. Thalamus is the nucleus, the area in the brain which um, combines almost all the sensory information, such as information from eyes and ears, and then thalamus transmits this information to the highest nervous center, to the cortex. So here we have the input from the thalamus, which brings here the sensory information, thalamocortical fibers. They are the axons of the neurons which are located in the thalamus. These fibers come here from white matter and go up to the layer 4. And here they end up upon the spiny stellate cells, which were seen here, spiny stellate cells. And then these spiny stellate cells innervate pyramidal neurons. And now we go to efferent fibers. Efferent fibers are formed with the axons of pyramidal neurons. So we have pyramidal neurons in layer 2, 3, and 5, and also some pyramidal neurons in layer 6. So we have association fibers made up with the axons of small pyramidal neurons in layer 2. Next, commissural fibers made with the axons of medium pyramidal neurons of layer 3. Next, projection fibers made with the axons of large or giant pyramidal neurons of layer 5. And finally, corticothalamic fibers made with axons of neurons of layer 6. And now we discuss where these fibers go, the direction of afferent and efferent fibers of the cortex. So, red color for afferent, incoming, and blue color for efferent, outcoming fibers. So, two types of afferent fibers, corticocortical and thalamocortical fibers. We discuss, for example, this particular area of the cortex. So this particular part receives information from some other portion of the cortex, and this input will be called corticocortical afferent input. And this area also has input from the thalamus. Thalamus is located somewhere here, not seen in this histological picture. It should be somewhere here. So we have the thalamocortical afferent here. And four types of efferents. The associative fibers made with the axons of small pyramidal neurons of layer 2 go to some other part of the cortex, but in the same hemisphere, which is called ipsilateral hemisphere. For example, here, this will be associative fiber. Next, commissural fiber made with axons of pyramidal layer of layer 3 go to other area of cortex, but in the opposite hemisphere, which is called contralateral hemisphere. Next, projection pathways, projection efferent fibers. They are made up with the axons of giant pyramidal cells of layer 5, and they go to motor neurons of motor nuclei of brain stem and spinal cord. So projection neurons go to the go downwards to the motor neurons. And finally, number 4, corticothalamic efferents. They go to thalamus. So we have here the loop between the thalamus and the cortex. You see that we have here input from thalamus to cortex, and we have back answer from the cortex to the thalamus. Now, the module. The cerebral cortex is organized in module. Module is the functional and structural unit of the cortex of the brain. The module is composed of so-called columns. Columns are the vertical structure composed of neurons of all the layers, starting from layer 6, layer 1 to layer 6, and the neurons which make one column in embryogenesis went along the same process of the radial glial cell. So these neurons form columns, one column, second, third, etc. And some columns are organized together to form a module. Module is for summation and comparison processes for informational processing. One module processes and integrates kind of one bit of information. For example, in the somatosensory cortex, one module may process the sensory information from one tiny area of the skin. Such modules are arranged in a so-called somatotopic manner, point-to-point correspondence of an area of the body to a specific point of the cortex. Here you can see the cortex and the area of the body which is the information from which is processed here. For example, this area will process information from toes. This area will process the information from hand, etc. Point to point. One point in our skin corresponds to a particular point in the brain. And this point in the brain will make one module, one bit of information. The somatotopic organization of the somatosensory cortex is illustrated in the so-called cortical homunculus, which shows the location of the receptive fields in our body surface and their corresponding location on the cortical surface. Cortical homunculus, which means small man, is a distorted representation of the human body based upon the neurological map of the areas and proportions of the human brain dedicated to processing sensory function for different parts of the body. The same type of organization of the cortex we have, for example, in the part of the cortex which processes information from ears, the auditory cortex. There we have the arrangement of modules for processing of sounds of different tones. In this picture we see again one module which has three basic compartments: afferent pathways, integration circuits, and efferent pathways. Here also different types of neurons are shown. So afferent incoming pathways include cortical fibers which came from some other part of cortex. These fibers go into the cortex and go to the surface, making small branches which innervate all other cells here. When they came to the surface of the molecular layer, they split into two and go along the surface. Next, thalamocortical afferent fibers which end upon the spiny stellate cells, and spiny stellate cells in turn will innervate the pyramidal cells. Efferent pathways include all the pyramidal cells of layer 2, 3, and 5, and also some cells of the layer 6. All other cells represent the integrational circuits inside the cortex itself, and all other cells are inhibitory neurons in the module, such as horizontal cells, axonic cells, basket cells, column basket cells, etc. You can see in this diagram what they do. The axons always make inhibitory synapses. Here you see the principle of organization of neurons in columns. So this is one column. It includes a small area of cortex, including neurons of all the layers. And the columns are organized in modules, such as here. And here is the organization of a module. Module includes one corticocortical input and two thalamocortical inputs. Here, stellate cells which process information from thalamus are shown in yellow. All pyramidal cells which make efferent fibers are shown in blue. They are triangular in shape. And other cells which are inhibiting interneurons, they are shown in black. So the efferent fibers of modules include association fibers, which go to 3 modules on the ipsilateral hemisphere. Commissural fibers go to 2 modules of the contralateral hemisphere cortex. Projection fibers go to brain stem and spinal cord, to motor neurons, and corticothalamic efferents go to thalamus and make the feedback from cortex to thalamus. Now, the histological preparation of the cerebral cortex. Staining silver impregnation. Once again, it looks brown. This is the sulcus, inside which we see the pia mater with blood vessels, and this blood vessel, as we see, goes into the tissue of the cerebral cortex. This is the cortex itself, it is the gray matter, and below the cortex we have the white matter. Now, you should be able to define the layers of the cortex. We have 6 layers. The most easy for us to find is to find the layer 5, because the layer 5 contains giant pyramidal cells, very large triangular in shape cells here. And first of all, if you are shown this picture, you should be able to explain why you think that this is the cerebral cortex. So the general feature of the cerebral cortex is the presence of the pyramidal cells, which are triangular in shape. And the layers are not very well separated from one another. So here we have the most large cells, triangular in shape. They are giant pyramidal cells in layer 5. So now we have found layer 5. It is here. So if this is layer 5, here is the surface, here is white matter. So just below the layer 5, in this area, we should have the layer 6. So we have found the layer 6. Next, we go upwards to the surface of the cerebral cortex and found once again triangular in shape cells, but smaller. These are pyramidal cells, medium in size, in layer 3. So this will be layer 3. Between layer 3 and layer 5, we will have layer 4 with stellate spiny cells, which receive input from thalamus. Next, we go to the surface once again of the cerebral cortex, and we should find the border between layer 2 and layer 1. Layer 1, molecular layer, uh, contains almost no cell bodies. It is filled with the nerve fibers. But in layer 2, we have lots of cell bodies. So it is easy for us now to see where is layer 2, where is layer 1. In layer 1, we see no cell bodies. In layer 2, we see lots of cell bodies. So now we have found all the 6 layers. Layer 6, then layer 5 with giant pyramidal cells, layer 4, layer 3 with medium pyramidal cells, layer 2 with lots of small cells, and layer 1 with almost no cells. The same is here. Another example of this preparation. This is the sulcus between two gyri. This is one gyrus, this is second gyrus. In the sulcus, we have here pia mater with blood vessels. Some of blood vessels are seen as white long spaces inside the cortex. Now, how to find the layers? This will be layer 5, because here we see here large pyramidal cells. This will be layer 5. Now we go upwards to the surface, and here we have found layer with the medium pyramidal cells. It will be layer 3. Then we go to the surface of the cerebral cortex. Here will be layer 2 with lots of small cells, and this will be layer 1 with very few cells. Layer 6 is between layer 5, where we have large cells, and white matter, where we have no cells, no neurons, only nerve fibers. Another example of this preparation, sulcus with pia mater with blood vessels, the cortex. In this specimen, we see quite well only layer 5 with giant pyramidal cells, triangular in shape. Their dendrites go upwards, their axons go downwards to white matter. Other layers are not seen, so we cannot say where is which layer, except layer 1, molecular layer, which is closer to the surface of the cortex, and it contains very few cells. The same for this preparation. Here will be layer 5 with giant pyramidal cells. Other layers are not very well seen, except layer 1, which will be here. Here at high magnification, we see the pyramidal cells, which are triangular in shape. They all have one long apical, which means going from the top of the cell, from the apex, apical dendrite, which go upwards to the molecular layer. And here we have basal dendrites, which arise from the basal portion of the cell body. They go to the sides to have the connection with other neurons. The apical dendrite goes upwards to molecular layer to find the afferent cortical fiber. So dendrites receive information from cortical fibers. So information will go downwards this way along the apical dendrite to the cell body of the pyramidal neuron, and next along its axon. Axon should be somewhere here. It is not very well seen, but it will go downwards toward the white matter. This is the neuropil. These can be the nuclei of the glial cells, not very well seen. So here, once again, the major feature of the cerebral cortex is the presence of the pyramidal neurons. All of them have one long, very thick apical dendrite, which receives information from above, from molecular layer. Basal dendrites to bring information from other neurons, and axon, which is usually very thin, which goes downwards to the white matter. Now we go to the peripheral nervous system. We will discuss the dorsal root ganglion. Dorsal root ganglion is the location of the pseudo-unipolar neurons. So this is the dorsal root. It goes to the spinal cord. Here should be spinal cord. In dorsal root, we have the area of thickening, seen also in anatomical preparation, more thick area here in the dorsal root. And it is thick because it contains cell bodies of pseudo-unipolar neurons. They are pseudo-unipolar because one process arises from the cell body, but soon it splits into two, one dendrite, which goes to the periphery this way, and one axon, which goes in opposite direction through the dorsal root into the spinal cord. So here will be the axon of the pseudo-unipolar neuron. In this histological preparation, which is called dorsal root ganglion, only this area, which consists round in shape cell bodies of pseudo-unipolar neurons, is actually the dorsal root ganglion. Other areas are not dorsal root ganglion, they have their own names. This is the dorsal root itself, this is the ventral root, dorsal root, here ventral root, and in this part they are combined together to form the spinal nerve. You should be able to explain in which area we have which processes of which neurons. For example, in this area between the neurons, it corresponds to this area in this picture, we will have both axons and dendrites of pseudo-unipolar neurons, which cell bodies are located here. In dorsal root, which is closer to the spinal cord, which will be here, we will have the axons of the pseudo-unipolar neurons. In the opposite portion, we will have the dendrites. Here will be dendrites of pseudo-unipolar neurons. In ventral root, we will have the axons of neurons which cell bodies are located inside the spinal cord. So, in dorsal root, we will have the axons of the neurons located in the spinal cord. And in spinal nerve, which is the combination between the dorsal and ventral root, we will have both dendrites of pseudo-unipolar neurons and axons of neurons of the spinal cord. So it is called mixed spinal nerve because it contains both axons and dendrites. And impulses in nerve fibers in the spinal nerve go in opposite direction. So impulses go this way along dendrites and this way along axons. This is the same preparation at high magnification. This is the dorsal root ganglion itself. This is the location of cell bodies round in shape, cell bodies of pseudo-unipolar neurons. Here we have nerve fibers. And this is the ventral root. It also contains nerve fibers made up with the axons of the neurons of the spinal cord. All the structure is covered with the dense fibrous irregular connective tissue which makes the capsule, which serves for protection of dorsal root ganglion. At high magnification, what we can see here, pseudo-unipolar neurons with their large nucleus and nucleolus inside, mental glial cells, they are cells which are located the closest to the cell body of the pseudo-unipolar neuron. For example, this is the mental glial cell nucleus here. Schwann cells and loose fibrous connective tissue, they are located more more far from the cell bodies of neurons. So these will be nuclei of mental glial cells. Those which are located more far, they will be the cell nuclei of Schwann cells and loose fibrous connective tissue. Between the neurons, we have loose fibrous connective tissue and nerve fibers with Schwann cells in their structure. And dorsal root ganglion is covered with a capsule of dense, irregular connective tissue, which is red in color because of thick bundles of collagen fibers. And these are nuclei of fibrocytes. Here we have some blood vessels in loose connective tissue, and adipose tissue may also surround this organ. At the most high magnification, we have here pseudo-unipolar neuron. Here is its nucleus, its cytoplasm. The nuclei which are the closest to the cell body of neuron are the mental glial cells. Others are loose fibrous connective tissue and Schwann cells. Now we go to the parasympathetic autonomic ganglion. Ganglion is the group of cells outside the central nervous system. We will look to the intramural ganglion, which can be found in parasympathetic reflex arc, and we will use the intestine as an example. So here we see the structure of intestine, lumen inside, villi here. Here digestion of food happens, and you see here the structure of the wall of the intestine. Intestine provides movement of food from the beginning to the end, and it happens with the help of the muscle layer. So in the intestine we have muscle layer, which consists actually of two layers, inner and outer layer. And between these layers we have sandwiched nerve plexus. It is located between two layers of muscles. In the picture, one layer of muscles, smooth muscles here, second layer here. They are at angle 90 degrees, so these cells are longitudinally sectioned smooth muscle cells, and these smooth muscle cells are cross-sected, and the ganglion sandwiched between them. Plexus, which means the network, is the combination of ganglia, one ganglion here, one ganglion here. In ganglia we have groups of neurons, and in plexus we have ganglia and connecting fibers. These are bundles of nerve fibers which connect ganglia together. So the ganglion contains cell bodies of neurons. Here important classification of neurons in the intramural ganglion according to Dogel. Dogel is the name of scientist who invented this classification. He classified the neurons into three types. Dogel type 1 neuron is the effector neuron. It has short dendrites and one long axon. Its function is to innervate smooth muscles and glands in the intestine. Type 2 neuron is sensitive neuron. It has long dendrites. It receives sensory information. For example, information about how stretched the intestine is in this particular site with a portion of food. And Dogel type 3 neuron is the associative neuron. It is the interneuron in this reflex arc. This is our histological preparation of the intramural autonomic parasympathetic ganglion. Intramural means that it is located inside the innervating organ. Here we see the muscle layer of the intestine, which is composed of two layers. Inner layer is here and outer layer is here. And The nerve ganglia are sandwiched between those two layers of smooth muscles. In the ganglia here, we will see the multipolar neurons. They will look black because of the type of staining, silver impregnation. Hematoxylin staining was used here also to see nuclei of the cells. Also, in the ganglion, we will have mantle glial cells, Schwann cells, nerve fibers, and loose fibrous connective tissue. The same preparation at high magnification. So these are cell bodies of multipolar neurons, types 1, 2, 3 by Dogel. Small oval and round dots are the cell nuclei of mantle glial cells, Schwann cells, and cells of loose fibrous connective tissue. We cannot differentiate between them, but those cell nuclei which are located the closest to the cell body of a neuron should be the mantle glial cells. All other can be Schwann cells or loose fibrous connective tissue cells. The ganglion has almost no separation from the surrounding tissues, so it has no capsule. Another example of this preparation, here we see three cell bodies of neurons, black stained because of silver impregnation. We have here their processes. We cannot say if they are axons or dendrites. This cell nucleus, very closely located to the cell body, should be the cell nucleus of the mantle glial cell. Other nuclei can be nuclei of Schwann cells or loose fibrous connective tissue cells. Those elongated cell nuclei stained with hematoxylin, they are cell nuclei of muscle cells because the ganglion is located between two layers of smooth muscles. Some smooth muscles oriented in this direction, as is seen from their nuclei orientation, and some in this direction. These are also smooth muscle cells. And now we will discuss the sympathetic ganglia. Intramural ganglia are parasympathetic. Sympathetic ganglia include sympathetic chain and prevertebral ganglia, for example, celiac plexus, superior and inferior mesenteric ganglia. We will look to this celiac plexus. This is the location of the third efferent neuron of the sympathetic reflex arc. This is how the celiac plexus looks like. This is the sympathetic ganglion in silver impregnation once again. It has the capsule, not seen here because of type of staining. And inside this organ we see black or brown in color cell bodies of multipolar neurons. So we will see here multipolar neurons, mantle glial cells which will surround these neurons, Schwann cells, nerve fibers, black in color, loose fibrous connective tissue, and dense irregular connective tissue which makes the capsule. These black lines are nerve fibers. So at high magnification we see here the cell body of the multipolar neuron, its processes. All the black lines here are the processes of some neurons. This is the nucleus of the mantle glial cell, because it is located very close to the cell body. Other cell nuclei are the Schwann cells or glial cells. Loose fibrous connective tissue cells. And finally, we look to the structure of the peripheral nerve in the cross section. Here in the left part we see the part of the peripheral nerve. It consists of bundles of nerve fibers. BNF stands for bundle of nerve fibers. Together, bundles of nerve fibers combine to form a nerve. A nerve is surrounded from outside with the epineuron. EPN stands for epineuron. Right part of the picture shows the same at high magnification. So here we see that epineuron consists of two layers. Outer layer is made up with dense fibrous irregular connective tissue, and inner layer, which goes inside and goes between the bundles of nerve fibers, it is composed of loose fibrous connective tissue. So epineuron, dense fibrous irregular connective tissue, and loose fibrous connective tissue with blood vessels. Bundle of nerve fibers contains nerve fibers inside. Here we see a part of the bundle of nerve fibers. These are myelinated nerve fibers. Somewhere here should be unmyelinated, they are not very well seen. Bundle of nerve fibers is surrounded with the so-called perineuron. PN stands for perineuron. It contains 5-6 layers of specific perineural cells. One nerve fiber, if it is a myelinated nerve fiber, it has the axonal cylinder in the center, the stained dot. It can be axon or dendrite. It is surrounded with not stained white myelin, which is made up with the um, wraps of membrane of the Schwann cell, and the cytoplasm of the Schwann cell makes very thin circle at the periphery, and in some cases the nucleus of Schwann cell is also present in the section. Between the nerve fibers we have thin layers of loose fibrous connective tissue with blood capillaries, and this connective tissue inside the bundle of nerve fibers between the nerve fibers is called endoneuron. E stands for endoneuron. So all the nerve is surrounded with epineuron. Bundle of nerve fibers is surrounded with perineuron. Between nerve fibers in bundle we have endoneuron. Nerve fibers, as you remember, can be myelinated or unmyelinated. In both cases, we have in the structure of nerve fiber an axon or dendrite of the neuron and a Schwann cell in the peripheral nervous system. So Schwann cells are present in all cases. But in the case of the myelinated nerve fibers, Schwann cell wraps many times around the axon or dendrite and forms many layers of its membrane, which is called the myelin sheath, because it contains of membrane of a cell, of a Schwann cell, and the membrane contains phospholipids. Lipids are stained with osmic acid. So lipids are stained with osmium in black color. So we use osmic acid or just osmium to colorize lipids in black color. So it looks black. This is how unmyelinated and myelinated nerve fibers look like in the transitional electron microscopy pictures. So in the left picture we have only unmyelinated nerve fibers with round, lighter, stained, cross-sected axonal cylinders surrounded with cytoplasm of the Schwann cells. In this case, the nucleus of Schwann cell is also present. This is the myelinated nerve fiber. Here is small unmyelinated nerve fiber. These are axonal cylinders surrounded with cytoplasm of a Schwann cell. Here, one axonal cylinder surrounded with a myelin sheet, very black in color, and the cytoplasm and cell nucleus of the Schwann cell. So this is our histological preparation of the peripheral nerve in cross-section. It is surrounded with epineuron from outside. It is dense fibrous connective tissue and loose fibrous connective tissue with blood vessels. Bundle of nerve fibers surrounded with perineuron. Inside we have myelinated and unmyelinated nerve fibers and endoneuron between them. So epineurum, dense fibrous and loose fibrous connective tissue. Perineurum, perineural cells, flattened. Endoneurum, loose fibrous connective tissue which brings here blood capillaries. Another example of this preparation, and some diseases of nervous system related to meninges and neurons and nerves. So this is all information. Hope to see you in class and wish you all the best. Thank you for your attention and goodbye.
Goodbye, Professor.